Well, good morning and welcome to the October session of ASU Health Grand Rounds. I'm excited about our topic today and like to welcome everyone who is visiting with us online and in person here today. Having a wonderful speaker, Dr. Hauptman, talking about tackling mTOR from bench to bedside, targeting genetic approaches to treating pediatric epilepsy. My name is Dr. Kristen Will. I'm Associate Dean for the Advancement as well as Executive Director at ASU Health, and excited about our session today. With that, I would like to just do a couple housekeeping pieces that will be offered for continuing education. And our speaker today did not have any disclosures. And just a reminder, these are the accrediting offerings for the interprofessional disciplines that we offer. So if you're desiring continuing education credit, at the end there will be an evaluation that you can complete and you will receive your certificate. Also, I'm pleased to say that we do have refreshments afterwards in the patio, so please join us for that. Students, you'll be rushing off to class, but please grab a bagel or some sustenance on your way out to start your day and sustain you. So we're excited about that as well. And just a reminder, these are the ASU Health Grand Rounds learning outcomes. Reflecting on that in your evaluation, these are here for us as a reminder. So with that, our speaker, Dr. Jason Hauptman, who's a professor of neurosurgery at Hoeye, surgery, a professor at University of Mayo Clinic, and of course at Phoenix Hospital. Hauptman is a nationally and internationally recognized expert in the use of robotic stereo electroencephalography, awake surgery in children, interstitial therapy, functional hemispherectomy, and neuromodulation, including deep brain stimulation and responsive neurostimulation. Before joining Phoenix Children, Dr. Hauptman served as an associate professor of neurosurgery and helped direct the surgery epilepsy program, the hydrocephalus program, and the tuberous sclerosis complex clinic at the University of Washington and Seattle Children's Hospital. So thank you so much, Dr. Hauptman, for being here. I will turn it over to you.
Thank you, Kristen. Everybody can hear me? Karaoke? I've been told I'm a baritone, so Neil Diamond is my go-to. Most of you have no idea who Neil Diamond is. You're not worse off for it. He was a star in the 80s and late 70s, but a crowd pleaser nonetheless. My name is Jay Hauptman, and thank you for having me. It feels really good to be at a university. I haven't stepped foot at the university in about a year and a half. Phoenix Children's is downtown, and when I was in Seattle, relationship with the university, since I was on campus all the time. And the reason why that's special to me is that there is always this sort of vibe of learning and advancement that happens really only at universities, hospitals. We try to recapitulate clinical care, a lot of other competing interests. And so it feels good to be surrounded by people who are here to learn, who are interested in cool stuff. Because I'm interested in cool stuff. So like Kristen said, I'm a pediatric neurosurgeon, career taking care of children with epilepsy. It's so bad that surgery is indicated, and brain tumors and some other things. And I'm happy to talk to you about all that. But when I sort of thought about what I wanted to talk to you guys today, I want to take you in about hopefully 35 minutes, a 10 to 12 year journey that actually just ended with a publication that is months ago. And it's been probably my most consistent career arc. You know, when you look at people who are academic in medicine, many people will have a variety of different research interests, projects that come and go, publications that are all over the map. You know, if you PubMed, you see things all over the place. But my career arc has been pediatric epilepsy, and specifically in the mammalian target of rapamycin. Started when I was getting my PhD. Now I've got to do the math. 17 years ago. Yeah. My knees are telling me it was 17 years ago. Most scientific talks you'll ever go to, they save the acknowledgments for the end. I hate that. I hate that. Everything I'm about to show you, I didn't do on my own. I wasn't even close to doing on my own. I had 30, 40, 50 people helping me. And these are them. Many of them are from Washington and Seattle Children's Hospital, colleagues from UCLA training me for PhD. One of the things that's beautiful about academia is you are who you are for the people whose shoulders you stood on. There is nothing I did that I did on my own, and there is nothing I could have done without these people showing me how to do it. Okay, and so I always like to humbly recognize all my collaborators. They are them. I'm going to really start at the basics. They aren't that basic, but the basics in pediatric epilepsy surgery, which is what I do for a living. When we have children who have epilepsy so bad that they require surgery, it's more than you'd think. Epilepsy affects 1% of the population. There's about 8 in 1,000 children diagnosed with epilepsy. Do the math. This is Phoenix. Talking about 10,000 kids or so. Okay. 30% of those children will have epilepsy despite medical therapy. That is a number. And it is incredibly taxing on the healthcare system. These are children who are constantly being admitted to the emergency room because they're in what we call status epilepticus. They're missing school. Their parents are missing work. Not to mention the effect the epilepsy is having on their brain development, brain health, affecting their developmental trajectory, and there's a risk of death associated with epilepsy that's 1% per year cumulative. Okay? Of those kids that come and see us in neurosurgery, by far and away, the most common problem we see is something called cortical dysplasia. Has anybody heard of cortical dysplasia? I think a few people in the audience have. Okay? Cortical dysplasia, for lack of a better word, is a birthmark of the brain. It is a series of precursor neurons that went away during development. And they wound up spun around, pointed in the wrong direction, dislaminated. They're in the wrong spot. You have like neurons in white matter. The problem. One of the symptoms of cortical dysplasia is epilepsy. With cortical dysplasia, we remove it. We resect it. Okay? But here's the problem. It only works. So there's a surgery that we can do. These kids need surgery. They need a solution. But it only works 50% of the time. So what happens when you failed medical therapy, you failed surgery, and now you're out of options 50% of the time? What happens to those children? And if there's something that surgery can only help 50% of the time, as it pains for me to say as a neurosurgeon, is it really a surgical problem? Or is surgery just the kitchen sink we're throwing at a problem we don't totally understand? I'd say it's the latter. So in the first half of this, I'm going to lay some groundwork and some basic science for those of you who love cellular neurophysiology. I was at one point in my career a cellular neurophysiologist. And I'm going to show you, I hope, how we turn that from the bench to the bedside, which is why I wanted to give this talk today, because people talk about bench to bedside all the time. I thought it was impossible when I started this journey. It was hard. Don't get me wrong, but it's not impossible. And for those of you in ASU bioengineering, when you're sort of fantasizing about how you're going to impact a patient, it's not as hard as you think. And I'm going to show you that's not as hard as you think. It's why I'm here. It's why I want to talk to you. It's why I want to meet you. All right? Because the physicians, like my partner over here, Dr. Chiarelli, is one of the neurosurgeons who works with me. We want to work with you guys to improve the lives of our patients, to improve the efficacy of what we do. So, mTOR, like for those of you who've ever seen Zoolander, mTOR is like Hansel. They're so hot right now. I think probably people over the age of 35 are going to get that reference. I don't. Has anybody under 35 seen Zoolander? Dude, that's a classic American cinema. I mean, yeah. I mean, that's that's Ben Stiller at his best. Okay, so mTOR is all over the place. It is ubiquitous. It's in every cell of your body. It's 289 kilodaltons. I just like to throw that out there. It's what's like a molecular genetics talk without molecular weight of an enzyme that, you know. And it's into a lot of different things: epilepsy, autism, TBI, brain tumors. It's all over the place. Of course, no molecular talk is complete without a picture of the mTOR pathway. But here's what's interesting about mTOR. Of all the different models we have of epilepsy that are in vitro, we probably understand the most about disruptions in the mTOR pathway because there is a prototypical disease that we see. It's not common, but it's not super uncommon, called tuberous sclerosis. Has anybody heard of TS? Okay. I see a lot of TS. It's something I've dealt with in my career quite a bit, and I've had some clinics where I take care of a lot of kids from all over the place with TS. We kind of understand TS. It is a downstream disruption in these two effector complexes because there's a deletion upstream of a regulator, TSC1 and TSC2. mTOR is just super, super important. It's cellular homeostasis, protein translation, cell survival, cytoskeleton reorganization and movement. And like I said, TS is the one that we understand the most. And in TS, these kids have epilepsy, they have intellectual disability, they develop brain tumors called subependymal giant cell astrocytomas. These are all big problems. And they have these things in their brain that are really similar to cortical dysplasia that we call TUBERS, not tumors, TUBERS, TUBERS. And that's like the pictorial, representation of what they look like. And these tubers, they cause epilepsy, very similar to the way cortical dysplasia causes epilepsy. This is what they look like on an H&E stain. But a lot of work done by my friends at the University of Washington, people I've worked with for years, and my colleagues and mentors, show that when we resect brain tissue of children with intractable epilepsy, and then we sequence that tissue, it turns out that probably greater than 50% of them have disruptions in that PI3K, AKT, mTOR pathway. And I'll tell you, it's more than 50% because for those of you in that line of work, it's all about depth of sequencing and how commonly this mutation is expressed in cells. What I learned a lot about is mosaicism clinically. What happens if this mutation only is expressed in 2% of cells? How hard do you have to look to find it? What's your false negative rate? Okay? And so we know that at least half of these kids have detectable mutations in this pathway. So to the lab we go. What does this mean? If mTOR is disrupted, if it's dysregulated, if it's going crazy, what does that mean for neuronal physiology? How does that translate into epilepsy? Hopefully I can show you an idea of maybe how it does. So we had a couple tools in the lab. One of them is a drug called rapamycin. It's been around for 50 years. Originally was used clinically to prevent rejection for kidney transplantation and actually was FDA labeled initially for the treatment of renal cell carcinoma. That's how it got into clinical practice. But for our purposes, rapamycin puts the brakes on mTOR. It very tightly binds to this complex and inhibits it. And it's very cheap and very easy to use. You can give it to animals intraperitoneally, and it will rise to very high levels both in the brain, CSF space, and in the body. It crosses the blood-brain barrier. Back when I started this work, this was cool technology. This is old technology now. This is the Phlox system of deleting genes of interest. In this case, we had an alpha kinase 2 promoter, which is expressed in hippocampal pyramidal neurons and frontal cortical pyramidal neurons. And in those neurons, you could knock out P10. P10 is upstream here of mTOR. So imagine a P10 knockout is the exact same thing as a kid with tuberous sclerosis, or in the case of our discussion here, cortical dysplasia. So if you knock out P10, mTOR goes wild. If you give rapamycin, mTOR is suppressed. That's like the simplest way of explaining the fundamental principles of what we did here. So we engineered these animals to have a P10 knockout and to have overexpression of mTOR. And it turns out when you do that, these animals seize. And they develop things that look awfully like cortical dysplasia under the microscope, with enlarged soma, enlarged dendrites, and about half of the neurons of their cortical pyramidal, hippocampal pyramidal neurons are affected by about 60 days of life in these animals. So we did a ton of electrophysiology. We tried EPhys just in normal animals treated with rapamycin where mTOR is suppressed. Then in animals in which we knocked out P10, in which mTOR was dysregulated. And then we did the rescue, which is where we gave those animals rapamycin to see if we could undo what the genetics did. Okay. How many of you guys are cellular electrophysiologists? Awesome, man. I'm going to put you all to sleep. It's great. It's been 20 years since I've done EPhys. So Dr. Andrews can unfortunately call me out if I say something incorrect. One thing that's about neurosurgeons is we're very sure of ourselves. So I may have no idea what I'm talking about, but it's going to sound like I do, because that's like a very neurosurgical thing. What the term we use is strong and wrong. Just because I'm speaking confidently doesn't mean I'm not messing up my electrophysiology. I'm going to try my best to remember it as best as I can. Okay, first off, when you take these animals and you delete P10 and you drive or dysregulate mTOR, the cells get huge. This is what they normally look like. This is what they look like when you knock out P10, significantly larger somatic area. That's very similar to what we see in cortical dysplasia when you look under the microscope, and the cells are big. The somas are big. They're very gnarly looking, dysmorphic looking, funny dendrites, funny apical dendrites pointed in the wrong direction. If you give those cells rapamycin, sure enough, they get small again. So I love that experimental model of a knockout rescue. My PhD mentor is a guy named Mike Levine, who's he's a dean or chancellor at UCLA, or was for many years. He wasn't when I was in his lab, but he was like one of those like old school electrophysiologist curmudgeon guys who I love, and he would hammer this into me, the necessary and sufficient experimental design. I love that design. It's very compelling. So what does this tell you? This tells you that mTOR, or P10, is both necessary and sufficient for driving somatic size, because you can undo what you did. Okay, so let's get into the EPhys for a second. For electrophysiology, we talk a lot about passive electrical properties and then what we call active electrical properties. Passive is how well a cell conducts electricity. Active is how well it fires an action potential. They're a little bit different, but they share some similarities. And here's what's important. What's important to take from this is that rapamycin, or mTOR, changes the way these cells conduct electricity. And it does it because it's shrinking these cells, and when it shrinks these cells, it reduces their capacitance and it increases their input resistance. And by increasing input resistance, you're amplifying signals. That's simple. You're making that signal louder. For a current change and voltage change, there's a significant change in amplitude, okay? Again, if you knock out P10, you undo it. You make these cells huge. You change the input resistance. And then you give rapamycin again. You make it small again. You change the capacitance. You change the input resistance. I'm not going to get into much more detail than that because I want to get to the cool clinical stuff in the time that we have. Just take my word for it that this pathway changes the way these cells passively conduct electrical current, okay? This is how they do it. And I think the message here is that by changing the way these cells conduct current, what you're really doing is you're either amplifying or dampening passively signals at the soma, okay? You have to shout louder to get that cell to hear you. Or the converse, you can whisper and that cell's hearing volume 10, okay? Next up, no good PhD thesis is a good PhD thesis unless you fail miserably the first time around, which is exactly what I did, because I thought this was all going to be glutamatergic neurotransmission. So I spent like months working on glutamate, came up with bookers, and then finally moved to GABA, and that's where the money was at. So thankfully it worked, but it took me a couple months of failure before it did. And so what I'm going to show you is that we just talked about passive electrical neurotransmission. Now we're going to talk about the synapse and neurotransmitters. And I'm going to tell you that mTOR changes the way cells hear GABAergic neurotransmission. This is what a typical voltage clamp recording looks like. In this case, we're knocking out GABA with bicuculin. I'm showing you the failure of glutamate. Nothing changes with glutamatergic neurotransmission. But when we went to inhibitory neurotransmission, we started seeing cool things. So this is what a normal cell will look like when you're listening for GABA. And this is what a P10 knockout pyramidal neuron looks like when you're listening for GABA. Significantly different, significantly reduced what we call spontaneous inhibitory postsynaptic currents, which is really GABAergic neurotransmission. These graphs are all demonstrating to you that this is real, okay? Now it turns out, like everything we've shown you so far, that you can undo it with rapamycin, necessary and sufficient. So again, suppress GABAergic neurotransmission in the P10 knockouts, that when you inhibit their mTOR activity with rapamycin, you restore GABAergic neurotransmission. Pretty cool. Love that stuff. It turns out that if you eliminate all action potentials altogether, remember that synaptic neurotransmission is not entirely action potential dependent. There's spontaneous release of GABA at the synapse. It's not AP dependent. So it turns out that when you knock out action potentials, that doesn't actually change the reduction in GABAergic neurotransmission. And it turns out that you can reverse this effect also with rapamycin. So this is with action potentials knocked out, you can actually improve GABAergic neurotransmission, meaning that reduction in GABA at the synapse is not action potential dependent, which is kind of interesting. We can also do things called evoked potentials. This is where you apply current in a different layer of cortex and then record from a cell in a deeper layer or more superficial layer. And it turns out that rapamycin also changes inhibitory neurotransmission when you evoke it. And when you knock out P10, you change evoked inhibitory neurotransmission. And it's rescued, again, by rapamycin. What do we take from this? Same thing. There's something about inhibitory neurotransmission that mTOR directly drives. And I'm going to just step back for a second and I'll put my epilepsy hat on and tell you that epilepsy is a result of an imbalance between excitation and inhibition, like in a macro sense. There's either not enough inhibition or there's too much excitation. So what do you guys think is the problem here in an mTOR knockout cortex? Not enough inhibition. Something is changing in the way cells are being inhibited. Okay. So from the basic science stuff, when it comes to synaptic excitability, again, mTOR is important when it comes to GABAergic signaling, and it's in a non-cell autonomous manner. Last of the electrophysiology, not only does mTOR change the way cells passively conduct current, the way GABA is acted upon at the cell surface, but it also changes what we call current clamping, or the way cells fire action potentials. So this is in current clamp, but the way cells fire action potentials. And what I'm going to tell you is that when you give rapamycin to a normal cell, you hyperpolarize that cell. You make it harder for it to fire an action potential. Again, when you have P10 knockouts, rapamycin does the same thing. It hyperpolarizes the P10 knockouts. It makes them harder to fire action potentials. And that's kind of cool, actually, because what that means to me is that even in a normal animal, rapamycin just suppressing mTOR probably has some sort of suppressive effect on the excitability of the neuron, which means in and of itself, rapamycin could be an anti-epileptic, irregardless of whether P10 is knocked out or not. Because by hyperpolarizing a cell, you're changing that delta between where a cell starts and where a cell needs to be to fire an action potential. So anyway, long story short, basic science altogether, which I'm really running through. mTOR is really important for the physiology of cortical neurons. It changes the way they act. It changes how easily or hard they are inhibited, and it changes how hard it is for them to fire an action potential. Okay. Any questions? I'm going to stop for a second. Any questions about the basic science? What I really hope you believe from me in the last 20 minutes is that this stuff matters in the lab. There seems to be a link. There seems to be something with rapamycin and mTOR inhibition and the way cells communicate. Would everybody agree? Would you buy that from me? Not just because I'm speaking confidently, but also like maybe some of the data looks a little compelling. Okay, cool. All right. Okay. Now, that data is old now. Okay. And I went back to my residency. I finished my residency. I did my pediatric neurosurgery fellowship. I went into the workforce. I did surgery, and I kind of let it go for a couple of years. And I worked on some other things. And then my soon-to-be chairman at the Washington, a guy named Rich Ellenbogen, and another guy named Jeff Ojman is my mentor in epilepsy neurosurgery. They invited me up to interview at the University of Washington. This is like a decade ago. And I'm sitting in their office, and they're like, so, if you come up here, what is it you want to do? Like, what do you, I'm like, I want to be a neurosurgeon. I want to take care of kids. They're like, yeah, of course, yeah. But like, what do you want to do scientifically? And I'm rattling off the 10 I worked on in the last 2 years, and they're like, yeah, that's all interesting, but tell me about this stuff you did like in the lab a decade ago. And I'm like, oh man, I don't know. I to set up a basic science lab. I don't know if I want to do electrophysiology again. Cellular neurophysiology is really for the true sadistic connoisseurs. Like if you really dislike yourself, like build a Faraday cage and patch clamp neurons. It's not the way to build self-confidence. And I'm like, I just don't know. Like if I really want to get back into building a lab and doing basic science again. And they're like, yeah, no, totally, man. But like, what you're showing us seems like it would work if you gave it to a kid who failed epilepsy surgery. And I'm like, whoa. This is like a lab. These are mice. Like, how do I do that in the clinic? Isn't that something like drug companies do? They spend like $5 billion. They take 10 years. They have advisors. They have a whole workforce of clinical trialists, and they're like, yeah, probably, but you got time, we'll pay you. Why don't you try doing it? In fact, we'll send you to a course over at the University of Washington in clinical trial design. She's never done that before. And so I was like, all right, and so I took the job. I went up there and I did exactly what they asked me to do, and I found out that bench to bedside is not as hard as you think it is. It's hard. A lot of steps, a lot of regulatory steps, a lot of things I knew nothing about. I had to learn the hard way. And it's expensive. It's expensive. This clinical trial I'm going to show you cost us about $1 million. And it was small, okay? But it was cool. So there was this company based out of Los Angeles. They're called ADI. They were called ADI. I think they changed their name. And what you'll find in the biotech startup is that there are a lot of small companies that have a really cool idea that are looking for an indication. So they need clinicians because they're like, we have this idea, we got this drug, we think it might work, but we need people to show us how we can use it clinically so we can get it through clinical trials and get a labeling indication. And that's where clinicians can be very, very effective. So this company, AADI, they had this drug. It was a rapamycin-like drug. It's an albumin-bound rapamycin, and they wanted to bring it to market. And I don't know why, but the thought they had bringing it to market was to bring it into a very rare genetic mitochondrial disease called Lee syndrome, which is very rare. I mean, there's Lee, and they wanted to bring it to market that way. And I was like, yeah, you can do that if you want. It's going to take you about 15 years to recruit 20 kids. Or you can let me try this in epilepsy, and I'll have a clinical trial done for you in about 5 years. And they're like, okay, that sounds good. And it turns out that the FDA has a lot of enticing benefits for rare diseases, what we call vouchers, that can be an advantage to small drug companies trying to bring things like this to market. And so I convinced them the way I'm trying to convince you that this is a good idea. And because I'm a neurosurgeon, it seemed like they believed me. And so I designed a trial, a phase 1 trial, I and other people. When I say I mean we. And this is why we did it. So I'm going to bring you right back to the beginning where we started. Epilepsy is a huge problem. 4 to 8 out of 1,000 children are diagnosed with epilepsy every single year, and 30% of them will be medically intractable. And of those 30% who are medically intractable, meaning medicine's not working anymore, 50% of them will have surgery and it will fail. That is a large number of children. They are at the end of their therapeutic road. What can we do? We try this. That's what we did. Why did we do it? Because epilepsy is bad. It affects developmental trajectory. It leads to intellectual disability, severe impairments across many neurocognitive domains. It affects children over their lifetime. These are children who are unlikely to graduate from high school, college, have a job, hold a job, be independent in caregiving, and then their caregivers have to sacrifice a lot to take care of them, as I would for my child. And so there's a huge economic impact too. But the most compelling reason to fix epilepsy in children is the mortality risk of epilepsy. So how many of you guys knew before I said that epilepsy kills children? Good. I'm glad you know that. Most people don't. They think that it's inconvenient to have a seizure. It certainly is. But there is a risk of death of 1% per year cumulative over a child's lifetime. That means that a child who lives for 10 years with epilepsy, was born with epilepsy, lives 10 years with intractable epilepsy, has a 10% risk of dying at 10 years of age. That is whack. That is messed up. There are very few diseases outside the cancers and the cardiac diseases that carry a 10% risk of mortality at 10 years of life, and a 20% risk of mortality at 20 years of life. That's nuts. That's how old you guys are. You know, imagine living with epilepsy, sitting where you are sitting, knowing that you have a 1 in 5 chance of dying from your seizure. That is terrible. That is a problem. So, when we were dealing with the FDA and we were asking them to use this, what they thought was an oncology drug for epilepsy, they did stiffen up a little bit. There were a lot of very good questions that were being asked. But in the end, what was the compelling argument was this. These kids are at the end of their therapeutic road. There are no trials out there, at the time, there were no trials out there for children who had failed epilepsy surgery. You know, there's just this endless cycle of anti-seizure medicines that aren't going to work because we know that the definition of intractable epilepsy is once you fail 2 medicines, a third, a fourth, a fifth, it doesn't matter which medicine it is, it's not going to work for traditional anti-seizure. Okay, so I showed the FDA all this preclinical data that I showed you. There's a lot of other stuff too that really, really great scientists did. People at Boston Children's, people at WashU, that showed that this drug seems to reduce seizures in animals when animals are preconditioned to have seizures. And then there was a fair amount of clinical experience with this drug in the tuberous sclerosis population. The original indication for everolimus, which is a rapamycin sister, okay, it's the same drug as rapamycin, just packaged a little bit differently, was for those tumors I told you about, subependymal giant cell astrocytomas. But when they did those trials, they noticed those kids seized less. That's called the EXIST-3 trial. So they gave a rapamycin analog, an mTOR inhibitor called everolimus, and they found that there was, depending on the trough levels in the serum, between 40 and 60% reduction in those seizures. So that was very compelling to the FDA. They're like, oh, okay, all right. We've used it before. So that was the rationale that we used for this drug here to give it to children who had failed epilepsy surgery. A phase 1 typically is a safety study, so that's what we did. So our hypothesis was that it would be safe and tolerated, meaning like children don't discontinue the drug because they don't like the side effects. And my hope was that maybe we would see an efficacy signal. But these are small studies. I'm telling you, this is a small study I'm showing you, but it took 5 years to pull this off. Okay. The hope was that maybe we would see an efficacy signal. And so that's what we did. That's what this phase 1 study was. And so we took children, who failed epilepsy surgery, and we enrolled a subset of them who wanted to be enrolled. And at day 0, we kept them at their home medicines. We followed them for a month. We gave them a bunch of questionnaires and things. On day 30, we started them on this experimental agent for a month. We saw how much or how little they seized, and then we did a 3-month washout. Does that make sense? So they were on their home regimen for a month, and we did our experimental intervention for a month, and then we washed out that experimental intervention for 3 months. Intuitive? Good, okay. We did it at 3 different dose levels. These were going to be if they weren't tolerating it. We never had to do that. This is called a 3x3 design. This is very common in oncology. But we did 3 different dose levels here, and they were tiered. So we did 3 children at dose 1, and if everything went okay, we did 3 children at dose 2, and if everything went okay, which it did, we went to actually 9 children at dose 3. And this is how you do drug studies in children. You look for what we call DLTs, or dose-limiting toxicities, and something called the maximum tolerated dose, which is that third thing there, okay? And so this is where we ended, okay? We closed this study about 2 years ago, and it took us about 2 years to crunch the numbers, and we had to do a bunch of follow-up at the FDA. And we just published this in Epilepsia in July. July's edition of Epilepsy. It hasn't made the print, but you can PubMed it now. And it's open access, so I think you can see it. We had 3 children that we did at the low dose, 3 children that we did at the medium dose, and 3 children we did at the high dose. And then I actually blew up the high dose cohort to have an additional 3 children in it. And everyone stayed on drug. And I actually have about 5 years of open label data, meaning that we had a couple kids that had such a remarkable effect on this drug that the parents wouldn't let me take them off of it. So I had to petition the FDA for what we call expanded access or open label, and they allowed me to do it. And so I have a couple of children who have been on this drug for now, actually 5 years. This is actually been on this drug for 7 years. That's a little bit old, so 7 years. So I have 6 of them that have been on this drug anywhere between probably 5 and 7 years. All the adverse effects were pretty minimal. But what I want to show you is this. This is their baseline seizure frequency. This is what they did when they were on drug. And then this is what happens when you wash it out. There was still a persistent effect. It's dose dependent. The higher the dose, the more the effect. I want to show you this really cool thing now. It's this. This is it, okay. Because I'm running out of time. I want to have time for questions. If you look at P05 and P06, P06 is the red triangle. P06 is a girl who my partner had operated on about 5 years before I joined the practice in Seattle. She had to have a surgery called a hemispherectomy, in which half of her brain was disconnected because her seizures were so terrible that she was going to die of them. And so she was having about 100 seizures a day. She had her hemispherectomy, so half of her brain was removed. It worked for about 2 years. and then her seizures came back. Now she was seizing from the only half of the brain that she had. She had no surgical options. And we actually sequenced her. And she had an AKT3 mutation, which is upstream of mTOR. So I had met her. She was seizing approximately 80 to 100 times a day. She had to be fed through a tube. She had a tracheostomy. She was devastated. And her mom's life was completely dedicated to just taking care of her daughter. I enrolled on the study, and by 2 months she was seizure free. But sporadic, cool data points are interesting, but is it real? Well, we put her on the open label, and it seems to be pretty real. She's been seizure-free for about 7 years, I want to say now, on drug. Her mom will never take her off this drug. This girl here also had a mutation, an upstream mutation in the mTOR pathway, and he predated our genomics, so we actually don't know what he had. So I had a group of kids who had tuberous sclerosis. Those kids are also seizure-free on drug, and that's cool, but that's not as cool as this to me, because TS I would have predicted they would have been in a dose-dependent fashion seizure-free on drug. I could show you that data another time. But these are kids that don't have tuberous sclerosis. These are kids that have cortical dysplasia. Or in the case of the hemispherectomy, she has what's called hemimegalencephaly, which is the worst form of cortical dysplasia you can have. And so for the first time, we have a drug that works on cortical dysplasia specifically, and incredibly well if we know their genomics, to kids who have disruptions in the PI3K/AKT/mTOR pathway. And so this is sort of where the work is headed. We want to identify these kids, and we want to treat these kids. And to do that, it's going to require a lot of different work, including sequencing the brains prospectively of children with intractable epilepsy. But I'm going to leave you with this thought because we're almost at the time. This is what is really cool to me about this. All this is great. But in epilepsy, the traditional thought process has been to use medicines that alter the physiology of the neuron. Every single anti-seizure medicine works on a receptor-gated channel. And every single one, like since the beginning, since phenobarbital. Phenobarbital actually, the by Hauptman, believe it or not related to me, was he was a psychiatry resident, and he was living in the sanitarium at the time. But that's how barbiturates were discovered. But for epilepsy, but the oldest drug to the newest drug, they're all doing the same thing. They're acting on ion channels. They all work as well as each other. None of them have been shown to cure epilepsy. They just don't. Okay, and I'm sorry. None of them have been shown to cure epilepsy in these medically intractable kids. They don't. This is an example of what I would call a disease-modifying agent or a biological agent. And in oncology, we've moved completely in this direction. Reason, traditional chemotherapies, they act in a very macro way to suppress cell growth and cell survival and cell division. But they're indiscriminate, which is why their side effects are terrible, because they're working on your gastrointestinal lining as well as they're working on your tumor. Okay. The idea here is like where we've gone in oncology, where we're finding genetic mutations in tumors, and we're using what we call targeted agents to affect them. An example that's transformed the field is there's something called the BRAF fusion in low-grade gliomas, and there's a drug that we can use on tumors that are BRAF fused that has really no adverse effects at all. Like kids can lead a totally normal life on this agent, and it will stop the tumor from growing. This is the same idea. This is a mutation in the PI3K-AKT-mTOR pathway that probably exists in at least 50 or 60% of kids that are going to brain surgery for epilepsy that we have targeted agents for. But we've never thought of epilepsy this way. This is like totally new way of thinking about it, using biological agents to molecularly target disruptions that lead to epilepsy. So my hope really is to put myself out of business. I mean, like, I love doing surgery. I love doing neurosurgery. I love helping children who have epilepsy, but there are days where I'm like, there's gotta be a better way. You know, when I see the kids that come back who fail epilepsy surgery, I know there's gotta be a better way. And so put me out of business. Any of you guys, please help me. Like, this is where I want to be. This is what I want to be doing. And so with that, I will stop. Michael Levine was my PhD advisor. He's retired now, was a dean at UCLA, taught me everything I know about electrophysiology. Carlos Cepeda was his, I love showing that picture of him, by the way. For those of you who don't know Carlos, but that's Carlos right there, walking on the beach in a very posing manner. But this guy here, this is what I wanted to point out. So talk about the arc of academia. Joseph was an undergraduate at UCLA when I was getting my PhD. He is now graduating neurosurgery residency at Yale and actually may be coming back to work for me here. And that's 20 years ago. That's nuts. But that's the coolest thing about academia, these arcs of mentorship that persist throughout years and years and years and years. And then the last person I just want to point out right here is Gary Mathern was the OG pediatric epilepsy surgeon on the West Coast. He transformed the field. He's the reason I became a pediatric neurosurgeon and why I got my PhD looking at these things. His career was cut short some years ago. He had a middle cerebral artery stroke that changed his ability to have to do surgery, so he had to retire early. But again, you know, while I'm here to talk about science, I always talk about the power of mentorship. And I wouldn't be where I am today if all these people hadn't impacted me in some way, but particularly Gary, who I still see all the time and I invite out to visit. With that, I think I stopped on time.
You did. That was great. That was wonderful. Thank you so much, Dr. Hauptman. I think we've got some questions up here, Dr. Anderson, I believe. I'm running. This is for the Zoom. Yeah, thank you.
This is fascinating. Have you started thinking about, for the folks who have germline changes in the PIK3CA mTOR pathway, about intrathecal delivery of CRISPR-based therapeutics and other types of genetic surgery?
Yeah. I mean, the challenge, so we are in the era now of gene therapy. Delivery is easy. I can help you with that. The regulatory pieces are still, I think, trying to mature. I think the greatest barrier, honestly, is financial. If you look at the journey of Spinraza, which transformed spinal muscular atrophy, I think the cost per year is something on the order of about $4 million per child. And so we have to overcome that. But like, that doesn't take away from the fact that, yeah, I want to be doing this yesterday. And if we have a therapy, I can deliver it for you. Like, so just to get some background for what she's asking, are there ways to deliver precision neurogenetic therapy to the brain, to the spinal cord, to the CSF? There are. Spinal muscular atrophy, which was a universally lethal disease, has now become a chronic disease, like high blood pressure or diabetes, due to the invention of Spinraza, which is delivered intrathecally through a lumbar puncture. It's just incredibly expensive, but who cares? And it's saving lives. Unfortunately, there is a healthcare economic impact to it. There is right now an ongoing trial, also intrathecal at Boston Children's for Rett syndrome. That one's really cool. I'm very interested to see what those results are going to look like. There's another rare disease called AADC deficiency. which can be treated through an intracerebral injection in the basal ganglia. And so these were science fiction like 10 years ago. I think if I had talked about this at the grand rounds 10 years ago, I would have gotten snickers, the audience. We're way past that now. I think the challenge that we have is going to be scalability, cost, and there's a lot of bioethical questions around transgenics that still need to be answered, but I think we need to answer them, and we need to do it, right? So yes is the answer. It can be done. It should be done.
Yeah, it was an excellent talk, very inspiring. The question I have is, you mentioned early on in the pathology of things, the hallmark is enlarged cells. Is there a reason, an underlying reason for that?
Yes. So when you look at the fundamental physiologic effects of mTOR regulation disruption, it directly affects cell size, probably related to both maturation pathways, growth pathways, cytoskeletal pathways, all the above. When you knock out mTOR, the cells get bigger. When you rescue with rapamycin, the cells get smaller. We know that it is a very much a necessary and sufficient action. What's probably happening in cortical dysplasia is that these cells are being trapped in a developmentally immature state, which is not only physical, they're large, they're funny looking, they're pointed in a funny direction, but it's physiological too. One thing I didn't even get into here is that for those of you who are into GABA, GABA actually has a flip state where it's excitatory in its immature state due to a different subunit composition. So like what's really wild is like just because GABAergic neurotransmission is affected by mTOR, if it's trapped in the developmentally immature state, it may be that GABA is excitatory, which is kind of interesting. But the answer to your question is like we know that mTOR is critical in these processes.
Great. So thanks very much for a great talk. And I believed everything you said initially about rapamycin, but I'm an immunologist, and rapamycin has some effects on the immune system. So is there any does the immune system play a role at all in epilepsy, or what, and what are the effects of kids being on rapamycin for a long time?
Great questions.
IVIG or?
No, great questions. So fortunately, clinically, there's long-term data of both children and adults on immunosuppression for a long period of time because of organ transplantation. So we know it's relatively safe. Now, interestingly, I'm using doses that are much lower than what would be traditionally used to prevent rejection. So, you know, an order of magnitude, like 20 mg per meter square versus 100 or 80, which is what you would use in an oncology or deliberately immunosuppressed clinical state. What we found, and I kind of breezed through it, but what we found are that the AEs, from a biophysical standpoint, the AEs are mild. Epistaxis, I didn't see any, they talk about pulmonary toxicity, I didn't see any of that. The worst thing is that kids will get the stomatitis, which is ulcers around their mouth, just probably due to mucosal turnover. But that's the answer to your second half of your question. To the first half of your question is, we know that microglia clearly play a role in a variety of neurological diseases, including epilepsy. We poorly understand it, but we know there's a role there. And so I believe very strongly that microglia certainly play a role in the acquired epilepsies. Mesial temporal sclerosis is the most common surgical entity seen in adults with intractable epilepsy. We do temporal lobectomies for them. Why does their medial temporal lobe sclerose? It clearly has to be immunologically mediated, certainly from just repeated epileptic events. It's like scarring. So the answer to your question is yes, I believe there is a role there. There's a lot of other data I didn't show here that's really interesting. So there are models that are very commonly used in the in vitro study of epilepsy in animals. There's the canic acid model, the PTZ model, and the kindling model. all of which are either drugs or electrical stimulation you can do to an animal very early on in life to make it epileptic. Okay? They're guarantees. You give PTZ, you give canic acid. That is how you give acquired epilepsy to a rat or to a mouse. There are studies that show that if you take that animal at the beginning, before you did any of that stuff, and you pre-treat that animal with rapamycin, you just give it to them like a normal animal. You just give it rapamycin. And then you try to make it epileptic, you can't. Think about that for a second. Like you pre-treat them with an mTOR inhibitor and you're like zapping their brain trying to get them to seize and they won't. That is an anti-epileptogenic effect that's never been described before. Like there's no such thing, at least until now, of an anti-epileptogenic agent, an agent that can be given that will prevent the acquisition of epilepsy. And it's probably related to the same thing. And so these are really interesting questions that just haven't been answered yet.
Yeah, has potentially disease modifying agent. Is there any evidence, particularly of a change, eventual change in size of the tubers in the brain or the renal regions, etc.?
Great question. Everolimus will reduce things like angiomyolipomas and cardiac rhabdomyomas, which are tumors that you see in TS. Tubers, interestingly enough, do not seem to physically change. And I think part of that is they are frozen in a developmental state. They've already gotten there. And I think that any effects that everolimus is having on the microscopic level isn't big enough to change the macroscopic structure of these tubers. A lot of them are also heavily calcified. And so I think what ends up happening is the calcification itself retains the ultrastructure of the tuber. Now, that said, at high serum levels, it does change the physiology because 40 to 60% reduction of epilepsy in kids with TS treated everolimus has been observed. And very clearly, there is a serum trough level dependency to it, meaning the higher the dose, probably the higher delivery to the CSF crossing the blood-brain barrier, the higher the efficacy. But also interesting is that in those kids who have a reduction in their seizure burden, their tubers look exactly the same. So you're not changing the ultrastructure of what their brain looks like, but you're clearly changing the physiology of probably the neurons around the tubers. There's been a lot of debate in TS as to whether the epilepsy is being caused by the tuber or by the gliotic tissue around the tuber. As a surgeon, I will tell you it's the latter, because I need to resect the rind around the tuber to get kids seizure-free. So there is an interplay between the tuber and the normal brain tissue that's probably affecting.
Great. Thank you again, Dr. Hauptman, for a wonderful presentation. We can give a round of applause. Thank you so much. And thank you all for coming. This concludes our presentation today. If you are seeking continuing education credits, please use the QR code on screen to complete the survey at the end.